It is a great pleasure to be here in this important conference on bioenergy to talk about natural products. So I would like to thank the organizing committee and especially Glaucia. She is a coordinator of bioenergy program at FAPESP and also a chair of this important event that I'm great to be here to talk about natural products in this conference. I think it's very important because when you think in sugarcane, you are completely into the fascinating universe of natural products. The next one, please. We have some outline of my presentation. It's very small, so I would like to talk a little about the importance of biodiversity, some importance of we are in Brazil. You have a huge biodiversity, some example of natural products. You have working along of almost 40 years in Araraquara, Institute of Chemistry, São Paulo State University. And also I would like in the final to give some words about the NUB database that I believe is the most important products in natural products nowadays in Brazil. The next one, please. I love this cartoon. It was published by Farvosk in Nature, so long. And also, I like because it's a beautiful, and you see this carpet, that means the huge biodiversity that you have in the planet. And also, you see that among so many different kinds of species, we that are mammals are very small amount with compared with insects. However, we have the great power of destruction and distinction of the biodiversity that was constructed along so development that fascinated that everything starts by a sequence of reactions, reductions, and oxidations when you became so well organized that is figure. The second one, please. Now, when you talk about biodiversity, we have some example along of our history that can explain and show how the nature produce sophisticated molecular structure that nobody could figure. So nature is the most important and the most fantastic lab, chemical lab, where we can inspire to looking for hits, leads, and medicines, cosmetics, food supplements, and other materials that is so can bring so many benefits for humankind. This is a very nice example from biochemistry that you have very famous professor, professor, who isolated this kind of peptide from jararaca, a kind of venoms of jararaca that is a snake on our biodiversity. And Professor Sérgio Ferreira discovered this beautiful peptide who inspired the captopril that used until nowadays to regulate high pressure. And it's very important because you inspire nature to take just one piece of this huge molecule and to produce several important and beautiful medicinal chemistry. The next one, please. This is, I like this. I produced this 2 years ago. That is a timeline of some natural products that changed the history of humankind in nature. When you, when you see the evolution of the species and the evolution of us in the planet, you see that so long, around 18th, 4th, it was isolated morphine. And morphine, until nowadays, is a very sophisticated benzyl isoquinoline alkaloid used as very powerful analgesic. And you see, during this long time, you see several kind of molecules that was produced for nature and until nowadays is used. And I would like to highlight the last one that was in 2018, that is um, cannabis sativa, cannabidiol, that you know this the recent compound and drug approved by FDA. So from cannabis that in Brazil is used as drug, produce a very important medicine for humans nowadays to, to treat some nervous disorders, and it is used. The next one, please. This is the fantastic kind of biomes you have in Brazil. Several kind of biomes. So you have a fantastic biodiversity. Uh, you are on the most important. Around 20% of all angiosperm is located in Brazilian biodiversity, and you see several kind of different biomes, like Amazon, Caatinga, Cerrado, and also Atlantic forest. Atlantic forest is very important for us in Cerrado because you are in São Paulo, and in São Paulo you have Atlantic forest preserved. However, due to the process of monoculture, especially sugarcane, the Cerrado of São Paulo was almost completely destroyed. And you have a very, very, very genuine kind of vegetation. And this kind of vegetation that is named ecozones between Atlantic forest and Amazonia, you have explosion of biodiversity. Explosions of biodiversity mean also very sophisticated chemical, so very sophisticated biosynthesis to produce several different classes of compounds. The next one. This is a project that I have participated in São Paulo. I am a member of a CBFAR, that's a huge project in FAPESP, CEPID, and also ENCT, that is also a big project, involve all 17 colleagues in 17 states. Is a huge national project, and these two projects combine and they support our lab nowadays in Araraquara. The next one, please. The objective of our project, when you work on biodiversity of tropical regions as Brazil, we have focus on science. You have several, a huge field to discover new ideas on chemistry about biodiversity, mainly when you focus on ecology. That is a tremendous interaction, environmental, and the sophisticated lab that nature can produce different kind of compounds in order to equilibrate and to regulate this kind of species in several different ecosystems. Also, we focus on several kinds of natural products that can benefit if you have investment. So we are in academia, you looking for new knowledge, but knowledge on natural products can be useful to drug discovery, to cosmetics, and also food supplements. So we are focused to work on biodiversity and also to have a straight relationship with bioeconomy. The next one, please. This is the experimental design that you have in our lab. There's not too much attention, just to show to you how sophisticated that you have all chemistry, part to take the extract from some organism to transform extract, and this extract to have all chemical information in order to have several different assays, preliminary and more pharmacological and toxicological assays. And thus, when you have all this completely, you can identify some leader that you can have a proof of concept and then maybe patent or to close some collaboration with companies. The next one, please. Now I show just three examples focusing different kind of examples that you have in labs. This I think is very important because I don't know if you know one very important estuary was produced with the cover about the consumption of some products that santo daime that several people use like a ritual of religions. So it's very important because nature doesn't produce nothing for humans. Nature produces biologically active compounds to survive during the evolution, to regulate, to adapt to several uh, climate, to several environmental. So this is fantastic. And we and men use nature as pharmacological activity or some kind of religion. This is very bad because this nature can kill soon. And this plant that is Malpighias used in this ritual, we have developed several work that was work of master, PhD, and postdoc. And you did a report and sent to Ministry of Health. It's very dangerous because people can die when take these potions and use in Brazil Tetrapteris mucronata, because the tetrapteris that produce the hallucinogenic tryptamines, you don't have. You have Tetrapteris mucronata, that is the same taxon but different species, and this species is very toxic. One tryptamine that you see in the six is lethal. So it's very important to see the chemical composition, to pay attention of the population that nature doesn't produce anything for us and can be very dangerous for us. The next one, please. This also is a very nice work that you developed in is a project Sprint FAPESP with Professor Emily Hilde, direct from South Australia. Futures Industry is a very nice institution dedicated also to sugarcane. You have worked a lot in sugarcane and you design several chromatographic profile and also acoplate hyphenate with mass and you see several metabolomics of these several cultivars of sugarcane in Australia and also in Brazil. It's very important because you can have idea about how the the environment can contribute for some difference in this chemical profile when you change the environmental and you have this metabolomic pathway. This like a fingerprint of all chemical that this plant produce. You have several very interesting flavonoids, C-glycosyl flavonoids that is rare. It is very important to study chemical ecology. The next one. This is another example of natural products, completely different for the first one. This is very important because Brazil is very dependent of agrobusiness. And you have one plant, Malpighiaceae, that produce some kind of compound that is very toxic. And these compounds is like a invasor plant species that contaminate the agriculture. And sometimes you have uh, cows, because you also is based on this kind of very important. And so we identify the species that contaminate these fields where you have huge cows. That also is very important for our economy. Né? So Tetrapteris is very toxic, and we have this word. The next one that is very important is not the compound that you map all chemical metabolites of this plant. You see different classes of compounds, and it's very important because people try to have some mistake about this species. The species that is in Brazil in these fields is not doesn't accumulate monofluoroacetic that is very toxic, but we produce this kind of 13 that's a steroidal compound that is not cause the death of the cattle, but it's also very toxic. They cause some kind of with the cows can abort, and also the small cows can suffer several kind of fragility, and also it's very dangerous. And we map, you discover that this compound is very dangerous to the economy of Brazil. The next one, please. The next. This just to illustrate one compound that you have in collaboration with CNPq and also with colleagues in Switzerland and Geneva that to study vegetables in Brazil. The next one, some kind of fruits, because we evaluate fruits in Brazil, they evaluate fruits in Switzerland. The next one. And also this we can see that we map all chemical profile in very, very small and also green. It's very important to mention in bioenergy conference that in our lab is green. You don't use chloride or no toxic organ solvents. You use only ethyl acetate and ketone to isolate. And this is a very nice methodology to map all kind of compounds. And this is very important because we got this prize. We got a prize from Abikin. It was selected, this work, in 2015. We got the prize in Abikin because from this kind of bark that is put in the trash, is industrial trash in the Amazonian, we isolate a very, 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 very important flavonoid, a dimer of flavonoid, that you have several patents. And also, we showed to the Amazonian that the bark of one fruit is more important the, from the fruits that use it to prepare cakes, to prepare creams, to prepare sweets. And the barks sometimes has a very important chemical composition. This is one example, and we have this work. And now this work is one collaborates with Sunrise, is a German company, and now we license this with this company because this company is a very important multinational responsible for raw material of biodiversity all over the world. The next one, please. Now I am finished this work and I would like to talk because so long work on natural products, we have a very important and chemical profile of all species we study. So we decided that we should create a database with all compounds that you have isolated in lab, 640 compounds. We organize by structure, by molecular classes, and also structures, functions, activity, and you decided to organize into a database. The next one. So this is the layout of our Nube database. Information that you have online. The next one. Uh, all of you know about the American Chemical Society, the chemical abstract. The chemical abstract is the most important chemical database in the world. They were interested in our database because it's the first database organized and certified of Brazilian biodiversity. And now you have a agreement, a memorandum agreement in collaboration. And after this, we realized at the end we are so happy because you have the first database in Brazil with 55,000 compounds already isolated from Brazilian biodiversity, all cataloged in chemical abstract that you can use it in this database. The next one. Uh, is another kind of compound that I think is just to pay attention of you, peptides, because it's large molecules. The next one is just one work that you have published nowadays. It's a focus in my lab, small molecule and these big molecules. The next one, large molecules. The next one. And final remarks, I would like to finish that we have a very, very huge biodiversity, and natural products is a very, very important of chemistry for science, and also very important information and base for drug discoveries and other bioproducts. Uh, our lab has formed along of this time several scientific work, someone's patent, but I think it's most important nowadays is to create the first database on natural products of our biodiversity. So it's very important. And also, I would like to highlight the importance of FAPESP, the importance of Biota FAPESP is a program created 20 years ago to study biodiversity, and we are in this program since its creation. The next one, please. Without students as undergraduate, master, PhD, and postdoc, should be impossible to have to be here or to have results in our science. It's very important to work with this new generation. It's all-time inspiration. The next one, please. And thank you for your attention. And I would like to mention in Portuguese this that I think is very important. Meditar se só as nações fortes podem fazer ciência ou se as ciências que as fazem fortes, principalmente neste momento tão triste que estamos vivendo de pandemia. E aqui os meus sentimentos para todas as famílias vitimadas por essa tragédia neste país, neste momento e em todo o mundo. Muito obrigada. Thank you very much.
Good day, everyone. It's a great pleasure to be back here at BBEST. And I'm here to talk a bit about the relationship between biofuels production and wildlife management in multifunctional agricultural landscapes. I'm Luciano Verdade. I'm an associate professor at the University of São Paulo at Piracicaba campus in São Paulo, Brazil. Next slide, please. Well, the term multifunctional agricultural landscape is important because it gives us the sense that agricultural landscapes are not only used for biological production, but also for biological conservation. In other words, they have a primary mission of producing goods and services from domesticated species, and a secondary but also fundamental mission of conserving wild species and their evolutionary processes. But that would include what's been called ecosystem services, but also what's been more recently called as ecosystem services. Next slide. But what biofuels have to do with multifunctional agricultural landscapes? Well, they are produced as any other regular agriculture. So there is a certain dispute between biological production of either biofuels or any other commodity and wildlife conservation. But as a matter of fact, wildlife needs agricultural landscapes to be conserved. So, because in no country, the conservation units like national parks are operational enough to provide conservation of wildlife as a whole. So part of wildlife still needs agricultural landscapes to live, to feed, to prey, and to evolve. But on the other hand, agriculture still needs the wild to be sustainable. And there's a certain paradox in this relationship because the value of wild is global and millennial, whereas its cost is local and annual. So there's a problem between a discrepancy between value and cost. In addition, agriculture has intrinsic non-economic, intangible values like culture and history that should be considered as well. Next slide. So in this relationship, there's a certain intrinsic conflict because of a certain dispute in terms of land use between agricultural production or biological production and biological conservation. So as a relevant part of biodiversity lives on agricultural landscapes, we need them. We need to provide them ways to live in those agricultural landscapes. As a matter of fact, like I said, it would be impossible to promote integral biodiversity conservation only in conservation units like national parks and biological diversity reserves, even if they worked it perfectly, which they don't. Next slide. But this conflict tends to become an interdependent relationship because extant domesticated crops and livestock depend on wild genes of their wild ancestors to adapt to environmental genes like global warming, changes, like environmental changes, I mean, like global warming, new diseases, etc. However, the wild ancestors of those domesticated crops and livestock are among the most endangered taxonomic groups on Earth. So, from one hand, wildlife or biodiversity as a whole needs agricultural landscapes to be conserved. But on the other hand, agriculture needs the wild or the wilderness to preserve their wild genes that are vital for their sustainability. So they need the wild to be sustainable. Next slide, please. That's why we are still able to find some even top predators in agricultural landscapes or those dedicated to biofuels production, like the spuma in eucalyptus plantations. Next slide. As well as their prey. So we have wildlife in agricultural landscapes. It means that we have their food web. They live, they eat, and they depend on the resources they find in agricultural landscapes, which means that agricultural landscapes should be managed not only to production, to biological production, but also to biological conservation. Next slide. So therefore, it's proper to consider the idea of talking about wildlife management in agricultural landscape as well as we do for national parks and preserved or pristine ecosystems. But what really matters in terms of wildlife management? Wildlife management can be resumed or summarized as the decision making of intervention in populations of wildlife. So when should we intervene in some populations in order to keep them, in order to avoid their damage or in order to use them? So when we think that way, we can find only four, surprisingly a small number, only four alternatives of intervention in terms of wildlife management. And that would include also agricultural landscapes. The first category of intervention would be the necessity of increased depleted population. This is the basis of what we call biological conservation. And it's basically focused on endangered species. On the other hand, for some species, we need to find the maximum sustainable yield because they have economic value. So we need to intervene in their management to assure their sustainable use. On the other hand, there are some species that can cause damage to human activities and human health, for instance, and we either have to manage the conflict with those species or to control them or to decrease their sometimes booming populations. But on the other hand, most of the species are not endangered or economic nor damaging. So we should keep an eye on them to see as soon as possible if they change their status. But when we talk about biological conservation of endangered species, we're talking about, officially, we're talking about a few thousand species in the world. When we talk about sustainability, I mean, well, let's focus in Brazil, for instance. In Brazil, we have no more than like 4,500 officially is considered endangered species. But when we talk about sustainable use, we're talking about a few hundreds only. But when we talk about control, we're talking about a few dozens. So the number of species that can be object of intervention in those categories are relatively few in relation to the number of species that exist in the world. But therefore, when we talk about monitoring species, we're talking about a few millions. So it can be frustrating for a young wildlife biologist, but despite having not much glamour, monitoring is the most important category of intervention of humans in the wildlife in terms of management, because we should keep an eye on them to see if they change their status due to some human pressure. Next slide. But what really matters in terms of monitoring? Wildlife monitoring is being based on monitoring diversity of patterns. That's why we call biodiversity. It means to monitor the diversity of patterns. Like this graph at your left, when we display the relative abundance of a group of species that can be detected by a certain method, for instance. But on the other hand, it doesn't make much sense to keep monitoring diversity of patterns because they are molded by the complexity of processes, of ecological and evolutionary processes that we could call them as biocomplexity, like for instance the trophic process or the evolutionary process itself. In this particular point, temporal dimension is particularly important because it drives patterns of abundance and distribution. Next slide. So what exactly should we use as an indicator to monitor biodiversity or wildlife into agricultural landscapes where biofuels are produced? Plant biomass is a common concept for biofuel production because we consider biomass as the whole mass of plant material that can be used for the production of biofuels. But on the other hand, the native vegetation also can has, biomass, both plants and animals. And there's in an agricultural landscape, there is a discrepancy between the rhythm of biomass or the pulse of biomass between the plantation and the native vegetation. For instance, sugarcane can go from virtually zero biomass, zero tons per hectare from day zero of plantation to more than 100 tons of green mass in a few months, in a year, in a yearly cycle, for instance. Whereas the native vegetation fluctuates slowly between what's for us wet summer and dry winter. Next slide. So biomass is a good candidate to be used for monitoring wildlife and agricultural landscapes because we can get parts of biomass that can be used for production and for conservation. But on the other hand, a program of monitoring, wildlife monitoring, should be based in crossing scale analysis. We should be able to detect local processes, but also regional processes, which is only possible if we have a net of long-term sites in the field, all of them producing data that can be interoperable. Next slide, please. We also have interesting results when we, for instance, see the influence of agricultural management in wildlife biology, for instance. This is an example where the pre-harvest fire, use of fire in sugarcane plantations affect the abundance of rodents in the area, which is interesting because sugarcane can be a sort of good habitat for certain species of small rodents, which are the basis of the food web in those agricultural landscapes, in those landscapes. So if agricultural management affects their abundance, that would affect the whole food web in the landscape as a whole. So we should take that into consideration too. Next slide. In small scale, we are using scats. Scats can be very informative. They can tell us what exactly the animal are eating, but also we can extract DNA from the predator in this case, and to see not only what species, but what individual from what species it is. So we can have good demographic information based on scats. We can also have good information about, besides their diet, also their ecto and endoparasites, and even their hormonal fluctuation in terms of steroid hormones like testosterone, progesterone, estradiol, and cortisol. So we can relate sex and age and diet and stress and reproductive biology in small scale. Next slide. So we can mix results in both small and large scale. Next slide. Using different techniques like the scats and also biomass and its relation with biodiversity. So in here we can see the relationship between biomass of native vegetation and the diversity of birds and their abundance and their ecological, functional diversity and taxonomic diversity too. You can see that the data are slightly diffused, but their precision and accuracy are compatible with the precision and accuracy we have in field studies of wildlife in monitoring programs. Next slide. So we can have a monitoring program based on the simultaneous monitoring of agriculture and wildlife, or in other words, in biological production and biological conservation, in order to assure agricultural sustainability, including their agricultural values and also historic and cultural values as well. But we can also monitor biocomplexity, which means the complex of biological and evolutionary processes itself that mold biodiversity patterns. So that would be the concept of this idea of monitoring wildlife in agricultural landscapes, and that I hope can be useful in special for biofuel production. Next slide. Thank you for your attention, and thanks for all the financial agencies that helped us to carry out this study. Thank you.
Dear colleagues, my name is Cesar Schiesari. I am a professor of environmental management at the University of São Paulo, and today my presentation will be about the ecology of sugarcane fields. The debate regarding the environmental benefits of a large-scale adoption of biofuels has heavily emphasized reductions in greenhouse gases, neglecting other highly relevant forms of environmental change, such as biodiversity loss. However, we know that biodiversity is sensitive to a variety of physical disturbances, both in magnitude and frequency, and to a variety of chemical stressors that characterized anthropic land uses. So it is very important that we understand what is the biodiversity value of sugarcane and how to improve it faced to other forms of land use. We propose to investigate the environmental drivers and the demographic mechanisms by which species are added, replaced, or deleted across gradients in land use intensification. We have emphasized freshwater ecosystems, in particular small freshwater ecosystems, both because they allow sufficient replication for statistical power and because they are inhabited by species that are amenable to experimentation in lab, mesocosms, and field, so that we can experimentally try to tease apart the factors that are driving changes in community structure across this gradient. Our model freshwater communities are dominated by algae and macrophytes as primary producers, as tadpoles as amphibian larvae as detritivores and herbivores, and a variety of predators, which include insects such as dragonfly larvae, beetle larvae, aquatic bugs, and fish. We have conducted comparative sampling surveys across gradients in land use intensification and found that sugarcane fields are impoverished in biodiversity, both of vertebrates and invertebrates, when compared to cerrados and sometimes also pastures. However, what we also found is that there are species that are found throughout the gradient and therefore are capable of colonizing and inhabit and are exerting their function in sugarcane fields. Focusing on amphibian larvae, what we have found is that Dendropsophos minutus, Dendropsophos nanus, Sinax, Physalemmus natereri, Physalemmus cuvieri, and Leptodactylus fuscus are found throughout the gradient. Actually, if we plot the frequency of occurrence, that is the percentage of water bodies in which they occurred, and their per unit area biomass, we found that these species are not being significantly affected by land use, at least by means of this criteria. So Nayana Cândido went over and measured and weighed thousands of these tadpoles. And with this, we have calculated the body condition index, which is the residues of the biomass as a function of developmental stage. And what we have found is that tadpoles in sugarcane fields are actually as heavy as those found in pastures and native habitats relative to their developmental stage, which suggests that they might be doing well. Furthermore, some individuals found in sugarcane fields are very large relative to their stages, which prompted us to understand what were they eating and what was their relative nutritional value. So João Barcelos Leite, my student, selected 3 of these species and took them to the laboratory, where he fed them the decomposing leaf litter of cerrados, pastures, and sugarcane fields. And what he found is that the decomposing leaf litter of sugarcane is of sufficient nutritional quality for permitting tadpole growth, and this happens because sugarcane leaves are comparatively tender and therefore decompose fast, yielding high-quality detritus. However, what we also found is that there are occasional faunal die-offs in ponds adjacent to sugarcane field, as witnessed in this particular example. Therefore, there must be episodes in which populations could be entirely wiped out from sugarcane fields. So we hypothesized that this could be due for the employment of agrochemicals. And we manipulated pesticides and fertilizers such as ammonia and vinasse, exposing a variety of freshwater organisms to these compounds at realistic dosages. And what we found is that both pesticides and fertilizers can cause significant mortality and sublethal effects, including growth, development, and reproduction impairment, in organisms as diverse as amphibian larvae, dragonflies, midges, crustaceans, and fish. To better understand the ecological, ecotoxicological, and biogeochemical implications of land management for bioethanol production, we moved to large-scale experimentation and actually converted extensive pastures into intensive pastures and sugarcane fields. This is one of our experimental sites in Brotas, showing 15 0.25 hectare plots. The 5 plots in the front are extensive pasture, the 5 plots in the middle are intensive pastures, and the 5 plots in the back are sugarcane fields. Now, this set of experimental plots received 15 9,000 liters and 18 3,500 liters mesocosms, which are constructed freshwater ecosystems. The realism of these constructed freshwater ecosystems can be verified by comparison with a natural terracing pond found inside of sugarcane fields and that contain a variety of insects and amphibian larvae. So these experimental plots were subject to conventional land management, including harvesting, fertilization with vinasse, and application of the insecticide fipronil and the herbicide 2,4-D. As one might expect, the physical-chemical changes in these mesocosms were steep, in particular after the application of vinasse, which is represented by the red dotted line. So dissolved oxygens dropped to 0 in sugarcane mesocosms. In turn, conductivity levels increased 4-fold, to a large extent by the increase in carbon, nitrogen, phosphorus, and potash, which, after a few weeks, promoted a bloom in phytoplanktonic algae. Biodiversity responses were varied, at least in terms of abundances. Colonizing amphibian larvae seemed to be irresponsive to land management. By contrast, insects, and in particularly sediment-dwelling and benthic insects, suffered the strong consequences of land management, in particularly vinasse and secondarily fipronil. We can analyze the trajectories of the communities of these 3 different types of land management by means of an ordination technique. What we see here is that vinasse and fipronil promote strong divergence in sugarcane communities. However, over time, sugarcane communities converge to those of pastures via the arrival of new migrants. What we found then is that the biodiversity of freshwater ecosystems depends on the management of the surrounding agricultural matrix. We infer then is that improvement of land management practices can promote an increase in the biodiversity of sugarcane fields. However, we know that even freshwater ecosystems are not entirely isolated in space, but are connected to other freshwater ecosystems by the dispersal of organisms over the terrestrial landscape. So these networks of freshwater ecosystems are called meta-ecosystems, and they harbor meta-communities of aquatic and semi-aquatic organisms. The consideration of meta-ecosystem dynamics tells us that, actually, spatial context can be important in influencing, in modulating the response of biodiversity to land management. To better understand these problems, my student Rodolfo Pelinson led a field experiment in which artificial ponds were created at 30, 120, and 480 meters from a stream and its floodplain, which function as a source of colonists dispersing over the terrestrial landscape. So a view of these rows of artificial ponds or mesocosms can be seen here. So these ponds were colonized by our model freshwater communities. But not only spatial isolation was manipulated, but also it was fully crossed with a simulation in scheduling dosage of agrochemical intensification that follows the conversion of cerrado into pastures and pastures into sugarcane fields. 1/3 of the ponds receive no agrochemicals and therefore represent, quote unquote, our cerrado ponds and communities. 1/3 of the ponds received weekly additions of nitrogen, phosphorus, and potash, simulating the gradual dissolution of granulated fertilizers and therefore represent, quote unquote, pasture ponds. Finally, 1/3 of the ponds receive not only the weekly additions of NPK, but also a single pulse of fipronil and 2,4-D, and therefore represent our, quote unquote, sugarcane ponds. We then follow community composition and structure and ecosystem function over time. So the results will be shown by means of ordination plots. So in these ordination plots, each symbol represents one community. There are 45 communities because there are 45 ponds. And these different symbols are joined in ellipses that represent each of the isolation treatments. So what we see right away is that spatial isolation strongly influences community structure, and that distances as close as 120 meters are sufficient to cause strong effects on community structure. And this happens because of dispersal limitation. Not all organisms from the source wetland are capable of reaching these ponds that are at 120 meters. Once agrochemical application starts, then fipronil is found to promote a major change in community structure, with communities of sugarcane pods, quote unquote, in the lower left, strongly diverging from the other treatments because of the deletion of insect populations and dominance by amphibian larvae. The subsequent manipulation of 2,4-D had no further consequence to community structure, and after 120 days, spatial isolation and agrochemical still were important and still influenced community structure, even though the community differences become more and more blurred over time. In conclusion, as other agricultural crops, sugarcane fields are impoverished in biodiversity relative to forests and cerrados. Yet, sugarcane fields harbor a subset of tolerant generalistic species. These species are either highly mobile species that visit or traverse sugarcane fields, or fast-developing species that colonize sugarcane fields until physical disturbances and chemical stressors remove their populations. Now, recolonization of sugarcane fields depends on organismal dispersal, which is inversely related to spatial isolation. In fact, distances as small as 120 meters into sugarcane fields are sufficient to limit species colonization. Now, despite significant recent advances, if sugarcane is to fully satisfy its claim as the leading feedstock for the environmental sustainability of fuels for transportation, biodiversity must play a key role in the agenda of the sector. That is, the biodiversity value of sugarcane fields can and should be significantly increased, and there are different ways by which this can be achieved. First, it could be improved by improved land management practices. And second, it could be improved by the protection, restoration, and conservation of both in-field and out-of-field habitats in landscape management. Thank you very much.
Hello everyone, my name is Miguel Calmon. I work for WRI Brazil, and I'm here representing the Brazilian Coalition on Climate, Forests and Agriculture. I'm going to present here the role of silviculture of native species and R&D in a new forest economy in Brazil. First of all, let me start talking a little bit about the Brazilian Coalition on Climate, Forests and Agriculture. We are composed by a group of multi-stakeholders from the private sector, financial sector, civil society organizations, and academia. Our goal is to articulate and facilitate actions for the country to promote a harmonic, inclusive, and sustainable land use in Brazil by promoting dialogue, by developing proposals on consensus, by promoting advocacy, and by communicating our results to this society. Here, just an example of the diversity of partners that belong to the Brazilian coalition. We have more than 270 members. In 2020, we held more than 200 meetings through the dialogue forums and the task forces. We also had several meetings with the governments. We also have a significant feasibility, more than 1,000 mentions in the press, and 95% increase in the followers through the social medias. We also put together several manifestations and technical notes, and we also did a very important work on communication through newsletters, events, videos, and website. The way we work, we have a vision for 2030-2050. We work on the four main areas. The first one is to produce more and better through agriculture and forestry. The second one is to end deforestation. The third one is to create value and generate benefits through native forests. And the fourth one is to make state public policies feasible and build aligned and integrated economic instruments. So we also have under each one of those areas, we have the task forces. And what I'm going to talk here today is about silviculture of native species, which is part of this task force. So what is the role of silviculture of native species and R&D in a new forest economy in Brazil? Let me start saying that Brazil is considered a forest country. We have 500 million hectares of native forests, but we also have 8 million hectares of plantation with exotic species. So there's a lot of need to also work more with native species, especially for economic use. So another important point is that even though we are considered a forest country, we still have very high rates of deforestation. So as a result of this deforestation, we have at least 50 million hectares of degraded pasture lands with low suitability for agriculture, with a moderate to severe level of degradation. So those lands could be easily used for a new silviculture with native species. So the potential is enormous. It is also important to say there's a big global demand for tropical timber, even though the rates right now are not very significant. But if you consider the new scenario of a low carbon economy, that we can see a lot of high intensity materials being replaced by forestry products, as well as our other goals and commitments to reduce emissions from the major supply chains and also to remove deforestation, we can easily see that silviculture native species can play a big role, not only to supply the global and national market for tropical timber, but also to help major sectors fulfill their commitments. Here, just also examples of current commitments that involves restoration and reforestation. So first of all, we just started the new decade of ecosystem restoration. It means that we need to accelerate and increase the scale of restoration in this coming decade. We also have the Paris Agreement. A lot of countries made commitments to restore and reforest degraded lands as part of their NDC targets. Just in Brazil, the commitment is to restore and reforest 12 million hectares of degraded lands. We also have other global commitments such as the Bond Challenge. It has a goal to restore and reforest 350 million hectares by 2030. And finally, there's a regional initiative in Latin America called Initiative 2020 that has a goal to restore and reforest 50 million hectares by 2030. So all those goals will certainly have space to bring silviculture to play a major role to help achieve those commitments. When we started this project, this idea to really develop this new forest economy based on native species, we started a project called Verena. It's evaluation of reforestation with native species and agroforest systems. So the goal was to demonstrate if there was economic viability on using native species for economic use. So the first step on this project, we gathered many experts in Brazil. We visited 3 major projects in Brazil with silviculture, native species, and agroforestry. And at the end of this process, after 3 workshops, we came up with 12 enabling factors that would be required to promote a new forest economy based on native species and agroforest systems. So here we can see R&D is one of them, but we have several others, very important ones, such as develop the value chain, the markets, public policy incentives, remove illegality, increase the amount of investment and credit, also the economic modeling. So we need to work on them in order to develop this new forest economy based on native species or silviculture with native species. So what we also did, we needed to demonstrate if there was a real business on investing on native species and agroforest systems. So we mapped in Brazil at that time, around 2016, 12 cases in Brazil that we could really do a robust financial analysis to assess whether or not there was a case for this kind of investment, not only for investors, but also for the farmers. So at the end of the whole analysis, we concluded that, yes, investing on reforestation with native species and also agroforest gives a good return, comparable or even higher with the traditional investments. But there's also a level of risk that is poorly known because the new activity, the capital needs is high, the investment horizons are long-term, medium-long-term, and liquidity also can be low and medium. So we needed to come up with what would be the strategy to really mitigate some of those risks and make this even a much better business for the farmers and for the investors. So here you can see on the graph showing again return versus risk. When you compare the assets with native species, the returns are, you can see, high or even better than a lot of the traditional investments. And but also we needed to come up with, okay, if we have this kind of perception of risk and so on, how we could really mitigate, what strategy would be required to de-risk those activities and make silviculture of native species and agroforest a better business for investment. So again, here you can clearly see that R&D also became one of the top activities to really develop this new economy based on native species, but you also have several others from capacity building, training, public policy, market, seed and seedlings, natural capital, how we can better assess the natural capital. If you can see this graph here, it shows what was the impact on investment on R&D during the last decades on several crops here in Brazil for sugarcane, corn, soybean, coffee, eucalyptus. So it is clear that investments on R&D was critical to increase the productivity of those crops and forestry. So the same for native species would be required. As you can see, for native species, even though a lot of research has been done for native species, but not really in a very, let's say, robust way to increase the current productivity of native species. So we are still in a very stable productivity. So there's a huge potential to really increase productivity and to make this a good business for everybody. So based on that, we launched in 2016 in the Brazilian Coalition Climate Forest Agriculture, we kick off the R&D program for silviculture of native species. So the first thing we did, we went to find support to support a study on research gaps and priorities in silviculture and native species. So we were very lucky to get the support from the World Bank Pro4 to conduct the study. So we made a call for proposals. We contracted a consortium of universities and research institutes, such as UFSCar, Instituto Florestal, University of Southern Bahia, and Embrapa, together with WRI Brazil, to develop the study. So we organized several meetings, and one of them was a 3-day workshops that really brought together universities, research institutes, research companies, consultants, and NGOs. So we ended up with 21 institutions representing 8 states in Brazil on this workshop to really come up with what to be the gaps in terms of research in native species and also what to be the priorities. So the end result of this work, the study, was a publication in 2019 called Research Gaps and Priorities in Silviculture on Native Species in Brazil. This is public available. And of course, we ended up selecting a total of 30 species, 15 in the Atlantic forest and 15 in the Amazon. And also we received some question about why do we have so many species when we start a new R&D program for native species. And our question is that there's a lot of diversity in the Atlantic forest and Amazon in terms of the species. We also wanted to have a silviculture with a diversified number of species, and also we want to manage that species in a sustainable way. And also the market demands very different type of wood based on color, on the quality and so on. So we want to really be in a position to really access the market through this new economy. So as part of the result of the study, we also conducted several scenarios. First of all, we selected 8 themes to work on this R&D program. And so it goes from seeds and seedling, genetic improvement, cloning, management, wood technology, markets, zoning and policy, legislation. And we did a lot of scenarios to see, look, okay, if I reduce the cost, if I improve my seeds and seedlings, how much this would impact in terms of return from my R&D investment. So we also came up with a total investment of $7.3 million on this investment. And also we want to see what would be the return on investment if I invest on this R&D program. So we did a lot of scenarios. We came up with for every dollar invested on R&D, we will get a return of $2.4. And also want to see what would be the minimum scale to justify an R&D program for native species in Brazil. We came up with a scale of 10,000 hectares, which is a very small scale compared to the millions of hectares that we can really reforest in Brazil with silviculture of native species. So after the study, we work on the R&D program itself with the whole governance structure, with the funding sources, with where we would implement all the research experiments with the key partners. So we finally launched the program on April 13, 2021. And now we are in the process to really work on several activities to really move forward with the program. Though the program will last 15 years with result in the short term, medium, and long term. So the overall goal of this program is to promote the technological development for the implementation of a forestry economy with silviculture species comparable to the main agro-industrial sectors in Brazil. So we are really looking into make this a very important part of the economy of Brazil because the potential is enormous, as I already showed. We also want to, again, develop state-of-the-art technological development in different dimensions, from forest production, environment, and landscape and human dimensions. We also want to stimulate the development of an interdisciplinary culture related to sustainable production, consumption of product and service related to silviculture and native species. Also, we want to contribute to the implementation of multifunctional forest systems which aggregate production and biological conservation. So this is the governance, to give an idea about the governance of this whole program. So again, it's a lot of players and a lot of themes to work on this program during the last 15 years. And again, we have some priority action right now to implement the research areas, development of the database, and also develop minimum standards for the experimental design and R&D protocols. So here we also selected already 30 potential research areas to implement the program in the Amazon and the Atlantic forest, and 4 of them already committed to start the program immediately. We are just now working on getting the funding sources. We have a potential funding source from the private sector, from development banks, and also from the institution that provides support to research. So this is just one example of one of the products we also produce during this process to develop the study of the R&D for native species. This is a book based on experience of 40 years in the natural reserve of Vale Espírito Santo State. So here, thanks again for all the support from Brazilian Coalition. Thanks again for all the organizers of the BBEST to invite me to present this, and also a special thanks to all the donors and supporters of the Brazilian Coalition. And also, if you want to know more about our work, you can just follow one of those social media. And thank you very much for this opportunity. Thank you. Thank you.